Good afternoon, everyone. Hope you can hear me fine. So we're going to start the webinar now. I think everyone is pretty much online, but if not, you can always watch on YouTube anyway. So I will be doing this live, so I apologize in advance if there's any unexpected interruptions. So let's begin. So today's webinar is going to be about antiviral compounds. So we're going to be focusing obviously on coronavirus because this is what most people are interested in these days. But to get there, we first need to talk a little bit about HIV and influenza as our two model examples. So, let's see. Right, so this is a little bit of a summary of what we're going to talk about today in the presentation. So we're going to go through the life cycles of three models: HIV, influenza, and obviously SARS-CoV-2 that I may call COVID-19, even though the virus is called SARS-CoV-2. So we're going to talk a bit about how they infect cells and what their life cycle is to work out where we can target them. And then just a quick overview of some of the antiviral strategies that are already in place for these viruses, especially HIV and influenza. We're not really talking about hepatitis today, just for time. And then we'll talk about some of the current investigation that's ongoing for some of these antiviral compounds for COVID-19, and then a little bit of a summary and context of this. Okay. So just trying to get the thing to move. Right, so it's just a bit of a lag changing the slides for some reason. So this is just a quick overview of antiviral drugs. So antiviral drugs are a bit of a non-specific term really for any type of medications that's used specifically for treating any kind of viral infection. So there's a lot of different subclasses. Some people make about 13. It depends on your point of view. Some are specific for some viruses, some are general, can be used for multiple different virus types. Here you have a bit of a timeline of the development of some of the key agents. The first one was edoxyuridine from 1963, and you can see when different viruses were discovered and then how many different compounds have been used to treat those viruses. HIV obviously is one of the key viruses that have been used to develop these kind of compounds because it's one of the newer viruses, obviously a pandemic virus as well, and especially in the early days with a very high mortality rate, so very concerning. Of interest, you can see that influenza has a very slow evolution in terms of specific antiviral compounds. One of the reasons for that may be also because of the availability of vaccines for influenza. But one thing to take in mind when thinking about antiviral drugs is that viruses are obligate parasites, which means that they are completely dependent on the host machinery for some of their biology and life, which means if you're going to interfere with the virus, you might also be interfering with the life. So you have to think of a specific target, if possible, that is specific to the virus and not going to affect host cell activity or cause toxicity, for example. So here's just a quick high-level overview of the three viruses that we're going to go through. We're not going to go through the biology of these viruses, but just a few quick things. So the three models we have today, all of these are RNA viruses. They either have a positive strand genome or a negative strand genome. HIV, interestingly, has two copies of the positive strand, so it's not a positive-negative double helix in the genome. These are all envelope viruses, which is also pretty critical when you're talking about fusion events, early attachment and entry, for example. Of the three, HIV is the only integrating virus, which is also important, which means it has a different step to the other viruses that we will see later on, how important that is. And the site of replication is also slightly different between them all. Obviously, SARS and influenza are more respiratory diseases, whereas HIV is targeting essentially the immune system. So also important here, though we're not going into too much detail, is the incubation time of the different viruses before the first signs of symptoms will occur. So HIV, it's about 2 to 4 weeks when untreated before you will get flu-like symptoms, for example, and then you might get the diagnosis. Influenza can be as early as 2 days after being exposed, whereas SARS at this stage, or COVID-19, seems to be about 5 to 6 days, so it can be as long as 14 days. The other thing that's also of interest here is how long you are a risk of shedding or a risk of infecting other people. So here for HIV, you could assume that that's lifetime, though obviously if you have an effective antiviral therapy, you may have essentially no viral load and be ineffective. This is obviously the case for products like Truvada or PrEP, where you can use it as a prophylactic kind of treatment and you'll get essentially no symptoms and so on. So one of the things I wanted to highlight here is some of the new data for COVID-19, which suggests that maybe the period of shedding is a median of about 20 days, which is quite a long time, which may be one of the reasons why these lockdowns are taking quite a long time to have effect. But there's some suggestion you can even detect the virus up to 5 weeks after infection. Influenza is usually, you can have shedding or infection from a infected influenza patient 1 day even before you get symptoms, so you can be asymptomatic and spread the virus to a couple of days after you have symptoms. Okay, so this first slide here is the HIV infection cycle. So we'll just go through it a little bit quickly. So looking over here on the left, you can see the virus here in kind of purple color. So the first step is really attachment. So in the case of HIV, this is mediated by the envelope glycoprotein, in particular the GP120 subunit that usually binds to CD4 on the target cell. When it binds to CD4, this causes a structural change in GP120, and then this allows binding to the co-receptor, which is usually CCR5, especially at the early stage of infection, but can also be CXCR4. And once this co-receptor binding occurs, you use the GP41 protein to mediate fusion. So the envelope of the virus fuses with the cell membrane and it releases the nucleic acid and viral components into the cell. So you might have, for example, fusion inhibitors there that could block that process. At this stage, you have reverse transcriptase from the virus that then converts the genome into the proviral pre-integration complex. So the important thing here, obviously, is you're converting an RNA genome into a DNA intermediate, which is the pre-integration complex. This is then imported into the nucleus where the virus integrase enzyme will integrate that DNA into the host chromosome. And this integrated DNA is then transcribed into the viral mRNAs and the viral genome. The viral proteins are translated and they're assembled on the cell membrane. The virus then buds out, but actually the virus is still immature at this stage and requires some proteases to activate the virus to become a fully infectious particle. So that's HIV. This is the quick overview of the influenza life cycle using influenza A as the model, which is the main virus type. So in this case, the attachment is mediated by the hemagglutinin protein, the HA protein, which binds to sialic acids on oligosaccharides and the cell surface. So this then allows the virus to enter into the cell in an endosome. And this then, the virus undergoes conformational change in some of the proteins, which fuses the virus envelope with the endosome and releases the virus ribonuclear proteins, the VRNPs, into the cell. These are then imported into the nucleus. And then some of the polymerases, in this case, the RNA-dependent RNA polymerase or RDRP, will then duplicate the genome and make the different viral intermediates for the genome. And also this is where the transcription of the viral protein will occur. So once that occurs, then obviously you get export into the cytoplasm, you get assembly and budding from the cell to get a full a viral particle. The full maturation process also uses this neuraminidase protein, the NA protein, which also is required for maturation. So when we talk about influenza strains, usually we talk about the different hemagglutinin and neuraminidase proteins, so the HN types. So for coronavirus, still some things to be learned, obviously, about COVID-19, but the main coronavirus infection cycle is like this. Again, you have attachment and entry. So in this case, it seems the viral spike or S protein is binding to a surface protein receptor. In this case of COVID-19, it's the angiotensin converting enzyme 2 receptor. The virus is then internalized. This can be maybe in endosomes, but also some fusion may take place. This can depend on the cell type, the pH of the endosome, and so on. In this case, there is a positive strand genomic RNA. So that means it's instantly translatable, let's say, into the protein. So this is where the viral polymerase will come from. So the polymerase will then mediate transcription of the genomic RNA and also convert it into a negative strand, which is then alternatively converted into the genomic positive strand again, or into subgenomic positive strand mRNAs for viral translation. So obviously the genome is then assembled with the viral RNAs and then exported in vesicular to the surface and then exported out where it mediates more infection. So here the key is, again, you have an RNA-dependent RNA polymerase, much as you do for influenza for mediating these intermediate steps. The maturation here also requires two different proteins, the membrane protein and the nuclear capsid protein as well. So that's just a quick overview of the infection cycle of those three viruses. So if we look then at HIV-1, we can see some of the antiviral compounds that have been used for HIV. This is not extensive, this is not all of them, this is just some of the key ones. So just for time, I won't go through all of the specific details. It would be interesting to talk about Truvada, for example, but there's not too much time here. But what you'll see here is that different stages of the virus infection cycle can be targeted: attachment, fusion, and entry. Obviously, you can block the entrance. For example, Marovirioc is a CCR5 blocker that can be used to block entry. Unfortunately, these kind of compounds aren't often used to treat HIV because I think it's quite difficult to get them to work in the right kinetics, for example. There are some new ones around. For example, Oseltamivir has recently been tested in cases of drug-resistant HIV. The reverse transcriptase step is probably the most common step targeted in HIV infection. You have all the different nucleoside analogs, for example, and the non-nucleoside products that basically block the synthesis of that DNA strand from the RNA genome of the virus. You'll notice here that I didn't mark any of these compounds in green because none of these are actually being used for coronavirus at this stage. Obviously, reverse transcription does not occur in the same way with coronavirus because it doesn't have an integrating step, so it doesn't need to make a DNA intermediate. So looking at subsequent steps of HIV infection, you have some products that block integration. I've highlighted ivermectin here because that some in vitro studies we'll talk very, very briefly later about seem to maybe have some use in coronavirus, at least in vitro. You'll notice here as well, replication and assembly. There's no products that really target these steps. This is because of what I mentioned at the beginning, that probably these steps are too similar to normal cellular processes. So trying to target those may be too difficult because it will probably have some quite significant toxicity to natural biology. And there's also the final steps, which is budding and maturation. So there's also a lot of interest in these compounds like protease inhibitors, especially because they can inhibit the maturation of the virus. And indeed, Lopinavir has been used for coronavirus, as we'll see later. With HIV, there is also another subset of products that are used that are called pharmacoenhancers or PK enhancers. So these are products that are used with other products to basically increase the pharmacokinetic profile. So basically to increase the exposure or the AUC of these products. That gives you better control, and you can use less doses and so on, and it improves the safety. The two main ones that are used are Ritonavir, which we'll see later as well, and Cobicistat as well. This is quite commonly used as well. It's also worth highlighting that for HIV, one of the treatment strategies is what's called highly active antiretroviral therapy, HART, or more commonly combination antiretroviral therapy, or CART. This is basically because they use multiple products, usually two, for example, reverse transcriptase inhibitors with a pharmacoenhancer at the same time. One of the reasons for that is it reduces the risk of having reversion and escape mutants, for example, from the treatment. I haven't got time to go into details on the mechanism of that, but that's clearly quite a critical issue, not only for HIV, but potentially later for coronavirus, and indeed also happens with influenza. So here's a quick overview of some of the antiretrovirals used for influenza. You can see here at the top the neuraminidase inhibitors. This is obviously specific to influenza, much like the reverse transcriptase inhibitors, because neuraminidase is not part of HIV or coronavirus, for example. I've highlighted Oseltamivir because we will talk a little bit about that at the end of the talk as well. There is this class of compounds used to treat influenza called adamantines, which are not often used these days, and one of the reasons for that is because you do get a lot of resistant strains emerging from the use of those compounds. Okay. So that's just a quick overview of some of the products that are available on the market. Why highlight those? It's basically because with all the speed required to get a treatment for coronavirus, it's necessary to repurpose some of these drugs where possible. So obviously you can't just invent from scratch a new antiretroviral specific to coronavirus in the short time that's required to get a treatment onto the market. So this is a bit of a recap slide, but it also adds a couple of other things. This is the coronavirus infection cycle again. So you can see the virus here, that ACE2 receptor, and some of the compounds that are used at different stages. So here, camostat mesylate is apparently being tried to block entry, for example. Arbidol has been used as well. Downstream, there's a couple of other products. We've mentioned Lopinavir, and we'll refer to that a bit later. For example, there's some inhibitors of the RNA-dependent RNA polymerase that are further downstream, and obviously the maturation steps later. Obviously, a lot of people are really interested in this compound here, Chloroquine and Hydroxychloroquine. So this is a compound that is being used to block the fusion and entry steps of the virus. It works pretty much on the endosome by affecting the pH and so on of the endosome and blocking the exit of the virus from the endosome. So these are not always used for other viruses such as HIV, for example, but were originally developed as antiviral compounds, but have commonly used now for malaria, for example, and also lupus. But we will talk a little bit more about those two compounds later. Okay, so there's obviously quite a lot of compounds here that we can talk about, but I'm not going to talk about all of them because there's just not enough time. So what I thought was more sensible to do is to limit the discussion today on drugs that have been published in clinical trials in the literature in the last week or two, and also the drugs that come under the banner of this WHO Solidarity trial. So the Solidarity trial is an international clinical trial that was developed by the WHO, and you can see the details on the internet about how this works, and I'll explain a little bit about the design here on this slide. If you remember one of the last webinars we did, which was about the regulatory concerns for COVID virus, we mentioned that WHO had designed trials for vaccines and small molecules. So essentially, this is what we're talking about again here. In Europe, at least, the arm of the Solidarity trial that's underway is actually called Discovery, and this is being led by INSERM in France, but is actually a multinational trial, including Spain and some other countries as well. So basically the idea of this trial is to try and focus the development of antiviral compounds on those that are most likely to work. And WHO has done some screening in March based on the literature and some of the anecdotal evidence that's coming out. And they've identified 4 treatment arms that will be used in this protocol, and that will be compared to the local standard of care. So obviously that may differ slightly between countries, but this is the most ethical and simple way to get the trial up and running. So the 4 arms will then include this compound remdesivir, which was used for Ebola as well as some other infections, Lopinavir and Ritonavir, which are used for HIV, as mentioned, with the same combination being used also with interferon beta 1A as a kind of immune stimulus. I haven't talked about the interferons before, just to keep it simple, obviously. And obviously everyone's favorite chloroquine and hydroxychloroquine as well. So these compounds are all used with standard of care. So that doesn't mean you stop giving ventilators and stuff like that. You still do all the standard of care, but you just add these compounds into the treatment regimen for the patients. Okay. So the population that WHO would want to include in this study are adults, so no pediatric patients. That makes it easier from an informed consent point of view, but the most patients are actually in this group anyway. So any patient can be included basically if there is no obvious contraindication to any of these products. So basically the design works that when a patient comes in, they'll be randomized at the local site to one of these 5 treatment arms. So this is not a double-blind trial because that's pretty hard to do in this case because you'd need to blind all the products and so on. So there is some concern for a placebo effect in this trial, but obviously being a large national consortium type trial, this should probably be reduced over the whole pool of patients that they should get into the design of the trial. So the trial is also adaptive, which means that if there is some futility or if there's some issues with some of the treatments, they will drop out eventually and the randomization will be reduced to those groups that have been still showing some effect. If you remember as well from that last webinar, this is a very similar design to what the vaccine trial that was used for Ebola, for example. So the endpoint here is the use of ventilation and intensive care and also the date of discharge. So they will basically be measuring on a kind of ordinal scale, basically the severity of the patients, which is semi-arbitrary. So you have a scale from 1 to 7 usually, which is 1 discharge from hospital and 7 death, with the intensity of ventilation and other treatment in between. So they'll just be monitoring the time. Other measures may be done at the local site, for example, other biomarkers or other clinical outcomes, for example. But the main outcome that WHO is interested in is obviously this clinical outcome here. So all the data will be submitted to a centralized point. It's going to be reviewed by a global data and safety monitoring committee. And one of the reasons is to reduce paperwork. WHO will just collect, in principle, at the beginning, the informed consent and then receive the data from the patients. So we haven't got any data from this trial yet, but we're going to look at some of the data that's been published for some of these individual treatments and so on from the literature. So the first one we're going to have a look at is this compound called remdesivir, which is being developed by Gilead. So this was tested as well in the Ebola trial, and I think it was actually dropped out eventually of that trial because there was no sign that it was working compared to the other compounds. Okay, so this remdesivir is an inhibitor of the RNA-dependent RNA polymerase that we mentioned. So it's obviously important for synthesizing the viral genome because it's RNA and so on. There's a couple of interesting and slightly controversial regulatory issues around this compound. Apparently in March of this year, so only 1 month ago, Gilead was successful in getting an orphan drug designation for this compound for the treatment of COVID-19. They already have an orphan drug designation for this for treatment of Ebola. And apparently the reason they requested this was to bypass the FDA pediatric requirements, so the PREA legislation. So the PREA legislation basically obliges you to perform pediatric trials for the time of marketing authorization. But Gilead obviously didn't want to do that, so they got the ODD to try and bypass that because orphan drugs are exempt from the PRIA legislation. However, due to some backlash or perhaps due to other reasons, they actually rescinded the orphan designation for that compound. And one of the reasons they stated was that even though this legislation still applies, that they should still be able to get accelerated assessment of this compound if it does prove to be safe and effective for COVID-19. So the main trial that has been published on this drug at the moment is this paper from the New England Journal of Medicine from Grine et al., which was a compassionate use trial. So this was basically an open label trial with 61 patients where doctors would basically write to Gilead and say, we want to test this, or they had some in the hospital and they gave the information to Gilead. So there were 61 patients at the beginning, 53 of these were evaluable. It was a no control in this and obviously open label, so that means it's a severely limited clinical trial design. If you look at the figure here on the right, you can see this is the kind of scale we were talking about for the WHO, where each patient is given a kind of severity score at the beginning when they're entered into the trial, and then at the end of the trial they're given another score. So an improvement is considered that if you go from a stage 5 to a stage 3, for example, that's considered an improvement. So here you can see the numbers. They claim that 36 patients, so these ones here, the light blue ones, of the 53 showed an improvement of 2 points on this scale. Some of the severe patients showed a little bit less success than the less severe patients, which you might imagine. I'm not sure what the current numbers are, but my feeling is that the severe cases is still about 50% of death rate. So I don't think this data suggests necessarily that there is a good response in these kind of patients at this stage. But obviously there is very limited evidence here because there's no control groups, open label, limited patients, etc. There is some isolated reports that this drug has been used for, but this is one patient at a time. So it's not, well, in this paper at least, so it's not possible to give a conclusion on whether this product is working or not at this stage. That doesn't mean it's not worth continuing to investigate, but there is no clear evidence at this stage. So the other one, this is probably arguably the best designed trial so far that's been published. This is the products Lopinavir and Ritonavir that we mentioned earlier, which are used for HIV. So one of them is a protease inhibitor and the other one is a PK enhancer, basically. So there was some suggestion that this combination, or at least Lopinavir, was useful against SARS, although some of the evidence for that in the trial at the time in 2004 was limited as well. But this was one of the reasons to pick up on this product. So in this trial that was also published in New England Journal of Medicine in 2000 and by Cow et al. A few weeks ago, this was a randomized controlled open label trial in 199 patients. So they basically had a control group, which was standard of care, or the treatment group. So they had 50-50 division between the two groups. The primary endpoint here was the same, essentially, as the Gilead trial and what the WHO recommends, so a 2-point improvement on that 7-point ordinal scale. So what they found here was that there was no clear evidence of efficacy, basically. So there was maybe a slight tendency for an effect. You can see here, and also the hazard ratio as a pure number might suggest that, but obviously the confidence interval does not suggest that there is any strong efficacy from this product at this time. So they had measured the viral RNA, so the viral load as well, and they didn't see a significant difference between the two groups, maybe some small tendency at some time point, but not really any strong data. They also suggested that some of the severe patients had a better effect than the untreated group, but again, that's not statistic. There's not a lot of evidence to suggest that that was working. They also found 14% of patients couldn't finish the treatment schedule due to adverse events. So the conclusion of this paper at this time was that there was no clear benefits from this study at this time. However, that doesn't mean that more studies are not useful or that there is no potential here. So this is why the WHO is continuing with study of this compound. Okay, so chloroquine and hydroxychloroquine are the two most talked about drugs for treating COVID-19 at this stage. The mechanism of action here appears to be to block viral entry, so it inhibits glycosylation of the host receptors. It may interfere with the proteolytic processing and also endosomal acidification. There's some suggestion that it has some immunomodulatory effects as well. It may reduce cytokines and stuff like that as well, but there's no clear evidence of that. Here I've just presented a little bit of the in vitro data from this paper from Liu that basically just shows you at different multiplicities of infection, so different concentrations of virus as it were, and different concentrations of drug, whether you get inhibition or not of infectivity. And you can see that it does appear to work, but that's obviously just in vitro data. There is about 80 clinical trials ongoing or registered at the moment for use of these compounds in the treatment of COVID. So it's obviously of high interest to a lot of people. The one we're going to talk about a lot is this trial here from Marseille, which is really the only really published one at this stage. So this was a trial that was done in Marseille in the south of France, and it was led by Professor Didier Raoult and was published in this paper here by Gautret from this journal, the International Journal of Antimicrobial Agents, and it was published on the 20th of March. We'll get back to some dates in a minute. This trial has obviously been reported significantly in the literature for very many different reasons. If you have a look at it from a scientific point of view, it's extremely limited and extremely controversial, and we will go through some of that controversy at the end. Here's basically the data. One thing I forgot to mention, they do two groups. They have the hydroxychloroquine group, and they also, which is the blue line here, the control group, which is not really treated with anything or standard of care, and they've also added an antibiotic here in this green bar. I can't even remember which one it was now, but it's not super critical anyway. But here are some major issues with this clinical trial. It's a low sample size, so it's 26 patients originally. 6 of these dropped out due to treatment failure. So basically they decided just to analyze the 20 patients that were still there. There was no dropouts from the control group, which is obviously expected. They had a different source of control population. So the control population was basically some untreated patients from another hospital or from somewhere else. There was at baseline different ages between the two different treatment groups, which may have affected the outcome. So the primary endpoint here as well was change in the viral load. So this is basically the primary endpoint here. So they're basically measuring how much virus is detectable in the blood, and they show it goes down, and they're saying that that's fine, we've cured the patients. But anyone who knows clinical endpoints will know that this is just a biomarker, and it's not even a validated biomarker, and it means nothing. For example, I can have viral load because I have been infected and be completely asymptomatic. So what you really need is that ordinal scale or another real clinical endpoint. So what else have we got? So there was no safety or any clinical efficacy reported in this study as well. There's also some severe cardiac safety issues associated with these drugs. And indeed, there were some trials of this compound even halted because of cardiac issues that came up in those trials. So what's the story then? I mentioned dates. So here we have this paper came out 20th of March this year. Looking back at the history of this, there's a huge lot of controversy that has come out of this. Firstly, if you look at the history, Dr. Raoult, he was promoting the benefits of this drug as early as the 26th of February. So this is almost 1 month before he even published the paper. So obviously it's an open label trial. He has a bit of bias maybe there to make sure that that result comes out in his favor and so on. There are some other reports from the use of this compound. There is a Chinese paper that basically said maybe there was something, maybe not, very similar to the other studies that I've already presented to you. There was also a rebuttal of this paper that was published on the 25th of March by Molina et al. In this journal MedMal. So this is another French group that basically repeated what he had done, and they said it didn't work at all. The journal that published his paper, and it's obviously not the best journal in the world, they basically retracted the paper in early April and said the article does not meet the society's expected standard. So you can still find it on the internet, but even the journal is washing their hands a bit. So obviously this trial was picked up, put on social media, went around everywhere, and unfortunately it was publicized in the wrong places for the wrong people. So President Donald Trump picked up on this and decided that it was a cure and started telling everybody to use it, and everybody went crazy, of course, and started to buy stocks of the drug, and now that it's just gone out of control completely. Why he's done that is a question that will become clear, I guess, with time. Maybe it's just he's not smart enough to look at the data or analyze it correctly. Maybe he doesn't want to do that. There was a report from the Washington Post that linked some alleged financial and corporate links to Donald Trump and the drug suppliers. Whether that has any relevance or not is hard to say, because this is also a drug that is available as a generic, but because there may be government deals going on and so on, there may be some interest there for Donald Trump to try to make some money from that. So even yesterday and today, there is again stories coming out about the French President Macron, who has been visiting Dr. Raoult to also find out more about this, and he also is promoting this, as is Bolsonaro in Brazil. Apparently, Dr. Raoult has presented to Macron some data from a new study that he has with over 1,000 patients with a 91% success rate. There's no data available, and there was a story this morning that came out that apparently it's another uncontrolled open label trial and probably of zero scientific interest at this stage until a proper clinical trial is done. Okay, so this is just something that's come out of this, which I picked up from some of the media outlets. This is a tweet on the left that you can see from some Trump allies, which is quite sinisterly called the Trump War Room, and basically saying that President Trump is saving lives because he's making everyone have hydroxychloroquine now and so on, and that everyone who says it doesn't work is a traitor and all that kind of rubbish that goes on with partisan politics these days. But basically the message is right now there is no evidence at all that this drug does anything at all for COVID-19. There's no good trials done, and that is basically what we have to do. It doesn't mean it doesn't work, it doesn't mean it does, we just have to do proper clinical trials. But anyone promoting this kind of hype and disinformation is responsible for the consequences of that, which is not good. So now we're just going to go on a quick aside, but this is influenza again. So this is just a little timeline of seasonal flus and pandemics. So you can see here the yellow lines, this represents the strain of influenza that is around at any particular time. The red bars basically indicate when that new strain emerges because of some type of pandemic. So you can see here, there's basically a pandemic every 25 to 40 years, sometimes a bit less, where you have a new strain emerging. One of the reasons why a new pandemic strain may emerge is because there's a lack of immunity in the population, which is exactly what's happening with COVID-19 now, that it is able to spread unchecked, basically because no one has natural resistance. Obviously, when you get a pandemic, you get a resistant population and it just fades, let's say, into the background as an endemic virus, basically. So the main influenza strains circulating in humans at the moment are the H1N1. These are the hemagglutinin and the neuraminidase proteins, as we mentioned before, different genetic versions of those. So before 1957 or 1968, there was really H2N2 was the only one circulating. There obviously is limited data the more further back you go. The first pandemic, though, of the 21st century was not COVID-19. It was actually the swine flu influenza outbreak in 2009. So this was caused by a H1N1 variant, which was where there was no antigenic protection in the population basically before it emerged. So even though it's the same H1N1 that was circulating, it had an antigenic variant that meant that people were more susceptible to the swine flu. So according to the CDC in the US, in the 1 year from April 12, 2009, ironically, almost the same dates we're talking about now, there was about 60 million cases and about 13,000 deaths, which is quite a few still, but most people have probably even forgotten about swine flu now. Worth highlighting that the US deaths are currently over 28,000, so over double that in only 4 months. So we'll see how that goes on. Here you can also see what they expect from the patterns of emergence and re-emergence. So this is also important that usually all these pandemics have a second wave. So that's what a lot of scientists are deeply concerned about now, that all the lockdowns will come down, etc. But there's going to be a second wave at some stage where the redistribution of the virus. So why talk about this? Because it's worth just talking a little bit about Tamiflu as a way to close off, especially since we've seen this controversy regarding hydroxychloroquine, for example. So Tamiflu is the active substance is Oseltamivir. It's given as a prodrug, which is a phosphate form. So since about the mid-2000s, so even slightly before the swine flu outbreak, but also because of the swine flu outbreak, several governments decided to stockpile this drug. So Tamiflu and also the related one, Oseltamivir, which is intravenous anti-influenza compound. So at the time of the swine flu outbreak, more or less, George Bush, who was president at that time, he and Donald Rumsfeld, who was the ex-chairman of Gilead Sciences at the time, were allegedly profiting substantially from this, because obviously the government at the time, they announced $7.1 billion dollars of federal money would go into buying these stockpiles of this drug. And obviously Donald Rumsfeld, who was, I think he was the vice president at that time, he was obviously in a good position to make a lot of money on that. When the swine flu break did come out, a number of countries wanted to update the Cochrane review of Tamiflu efficacy. So anyone who's not familiar with the Cochrane reviews, these are reviews done by independent groups based on published clinical data to try and perform a meta-analysis to really show in a more objective way, do they or do they not work, these compounds. And what they found when they redid this Cochrane review was basically that Tamiflu does not work. They have a very small effect on reducing symptoms and can help some populations very little. Maybe you get 1 day less infection or serious infection compared to other non-treated patients, for example. And it can reduce in some way a little bit it can be used prophylactically, again, to a limited extent. But basically, the overall finding of the Cochrane review was that these drugs do not have any real capacity to do anything and do not have any impact at all on a pandemic, which makes you wonder why they bought all this stuff in the first place. But probably, again, it's a similar situation to the hydroxychloroquine quinolone. But what's also important here is that when they started this Cochrane review of Tamiflu to really decide if it was effective or not, it led to this long-running legal battle that is still going on. And indeed, you can see this headline here, which came out, I think it was from January this year. And it's from the Organized Crime and Corruption Reporting Project. And basically, there has been a lawsuit against the Roche, who was the original maker of this, for basically fraudulent sales because it wasn't actually doing its job. But one of the consequences of this Cochrane review was the battle to get actually the original clinical study reports that could then be used to make these Cochrane reviews, rather than having published papers that may have some bias from the pharma companies, for example. And this was the consequence of this was the change in the clinical trial transparency laws that many of you may have seen over the last couple of years, because basically there was no forced publication of clinical trial data to perform these kinds of analysis. So this is now a legal requirement, but I believe there is still a lot of issues getting big pharma companies to put data into the databases to overcome this interest. There was further investigations on this issue, and in 2010, the British Medicine Journal found that there was conflict of interest in scientists who were advising WHO at the time and recommending this drug for pandemic situations. And since then, WHO has downgraded this product, and basically it's not on the essential medicines list at all, because basically they say it doesn't work and its effects have been exaggerated, even if it does have a modest effect in some situations. So if you're interested in that story, I recommend to have a look at the BMJ website, where they have quite a lot of information, because they were the ones leading that investigation. And those who have read the book Bad Pharma will also remember that story. Okay, so here's just a quick recap of some other agents then before we finish. So we mentioned ivermectin before. This has been shown to inhibit HIV-1 integrase, but it also may inhibit the nuclear import of some viral proteins. So there was a paper by Kelly et al. That showed that it was potentially effective, but only in vitro. So again, not very informative at this stage, but again, possible drug. We know that coronavirus is entering via ACE2 receptor. So ACE inhibitors and angiotensin receptor blockers may have a potential role in the future. There is a lot of controversy around this as well, because in the original reports coming from China, there was apparently a lot of hypertensive patients who were in the more severe categories of COVID-19. Whether that reflects the fact that having these kind of drugs as a background drug, for example, for hypertension, whether that's causing an exacerbation effect or not was not clear. It's since come out that maybe the number or the proportion of hypertensive patients in the severe groups was not actually reflective of the whole of Chinese society, and it may have been actually less, which may mean that these drugs were actually protecting some people. So there is a lot of controversy there about whether you should keep taking these drugs or not, and more studies will be needed. There is also some other unsubstantiated reports looking at anti-inflammatory drugs or even immune promoting drugs. So the two main ones I've just focused on here are tocilizumab, which is an anti-IL-6 receptor antibody. One of the reasons why this was promoted in this Lancet paper was there seems to be a tendency or suggestion that the real severe cases of COVID-19 may result from a cytokine storm. This would be highly consistent with some things that have come out, for example, from gene therapy clinical trials with very high viral titers being administered, where you can get cytokine storms. And in fact, the Jesse Gelsinger case may have been the result of something similar. But also there is some neuropathy that's apparently being seen in some of the severe COVID cases that may be relating to the retrograde transmission of the virus from the olfactory nerves into the brain, for example, but also cytokine storms can have neurological consequences, as people who work on CAR T cell therapies will also know. And this compound is actually used exactly for that in CAR T therapies. Some people are also promoting the use of anti-TNF therapies because apparently TNF is very elevated in the blood and diseased tissues of COVID-19 patients. However, these have a risk because obviously anti-inflammatory drugs may mean your immune system is reduced a little bit, which means the virus may actually go out of control, or it might cause secondary bacterial infections. So these drugs at the moment are a little bit controversial, and more data is needed to see whether there is a mechanism that could be targeted with these drugs. Obviously, there's a lot of talk as well about convalescent plasma. So that's basically taking the plasma of patients who have survived COVID-19 and administering them to patients and using the natural antibodies of these patients to treat the condition. So this approach was done as well in Ebola and was shown to work. However, as a wide, large-scale approach or as cross-community approach, that probably is not going to be a feasible approach in the long term, but may help critically ill patients in the hospital. So here's just a few other considerations when looking at antiviral compounds. Okay, so the first thing is when you're starting off on these in vitro studies, which is the strain you have to use? Guidelines will tell you that you should use both laboratory strains and also clinical isolates to show inhibition. And indeed, that's what people do with HIV and so on. Quite often you'll see multiple in vitro experiments with different strains done. Also, there is usually a requirement that resistance is examined during the clinical program, because that's obviously a concern. And even in acute cases of influenza treatment, that has been observed. And in some cases, for example, hospitalized patients, particularly, the treatment can go on for up to 2 weeks, for example, which may be sufficient time to cause the emergence of resistance changes. So usually you have to look for genotypic variants and so on in the different isolates that you find. There is also a risk that you may cause cross-resistance to other drug classes. So this is not commonly shown, but it is a risk that has to be considered when applying these drugs. Another thing is the dose range finding studies. I didn't talk very much about which doses were used and so on in those trials, because basically they quite often use different doses depending on the patient, or maybe they don't respond to one, they might put it up, for example. So the question can be quite hard to answer about what is actually the effective dose to do here. So here, there's quite a few questions. So obviously we're repurposing drugs here from HIV or influenza. So the dose used for those viruses may not be the right dose to use for COVID-19, for example. So you may also have to use high doses in the case of resistance, for example. There's also the use of those pharmacoenhancers, for example. These may also cause drug interactions with other drugs that are being administered to these patients. So that also has to be considered as a possible risk. And obviously, if you're doing combinations, as some of the cases are, how do you titrate the dose of the individual components in those studies? Pediatric use of these drugs has not been considered very much at all, probably for 2 or more reasons. One of the reasons is obviously that adults are more severely affected than pediatric patients, although pediatric patients still get COVID-19 and some still have died. The other may be obviously getting informed consent and so on is much more complicated in those patients. Again, whether these formulations are suitable for pediatric patients is another question. There may be too big the pills, they may have some excipients that are toxic for especially young children, for example. So there's really a lack of dosing and safety data for a lot of these compounds. It's also worth highlighting that all those compounds are essentially oral use compounds, which is fine, but actually there is a need probably for intravenous formulations for very severe patients who are not able to swallow medication. And in fact, one of the pandemic influenza products was intentionally designed to be an intravenous use product for that purpose. Another thing to consider is obviously special populations. COVID-19 has a large elderly target population, so there may be different PK requirements, lots of co-medication going on and so on, so that can be another important issue. These patients may have renal or liver deficiency, so again, dosing adjustments may be required there. And some patients may also already have other viral infections. What happens to an HIV infected patient on CART, for example, who gets COVID-19? Or do they get COVID-19 because maybe the CART is already killing the COVID-19? I haven't seen any specific studies on that. Hepatitis B and C are obviously other huge considerations there. And obviously when designing these trials, the question can also be, when do you start giving these products? And what is the target population? Do you give only to severe? Do you give to moderates? How does that work? And obviously that kind of stuff will have to come out slowly with time. So some conclusions. Just to make it very, very clear, there's absolutely no evidence at present to recommend any specific anti-COVID-19 treatment for patients who have confirmed COVID-19. That is the WHO recommendation at the moment, and it's based on the data that I've presented to you. I have seen no other clinical trials that are published and no other data, and no data at all, as you've seen, suggests that any of these compounds is working at this stage. We've seen a lot of studies in vitro, which is fine, but that doesn't mean anything. That doesn't mean you have any in vivo efficacy. And indeed, historically, there have been some cases of compounds that have looked good in vitro and have actually exacerbated infections in vivo because they overcome a mechanism that's useful for the immune response or something that is needed. So extreme care needs to be taken there. So the current clinical trials that we presented in the talk today, these are generally not well designed, which is a consequence of the circumstance as much as the scientists behind them. So they are often not good enough to give meaningful conclusions, which is why the solidarity trial of WHO will be very important when we get the data from that. The data from that should be out within a few weeks, I imagine, but it will take some time. As we mentioned, the timing of the treatments and other clinical factors will also be very important to have a look at. So what we need to have here is basically if any of these compounds are to emerge and to emerge quickly and for the benefit of all people and not just for some people, for example, or some populations or some countries, what we need to have is a really rigorous community-wide study with high levels of standards and full transparency. So this is basically the WHO Solidarity Trial. We don't need some renegade scientists doing some random studies and then trying to sell and make a career out of making these antiviral treatments. Need some rational response here and some scientific objectivity. The final message I would give to you is basically, I assume that most people who are watching this will be scientists. So we have a kind of responsibility, I think, in these times to be able to communicate some of this information to people who are not scientists and to help in what we call objective health literacy in the general population to stop these kind of fake news and exaggerated stories coming out and so on, and to help people be able to analyze correctly the information that they are receiving, because there's too much misinformation going around at the moment. And indeed, the WHO has created a special task force and website to deal specifically with that kind of information. So I say that as well because I've had many people ask me about this compound who are not scientists, so it's good to be able to have some knowledge about this and be able to go to the papers and look at yourself and try to communicate to people to give them a clear idea. And the clear message at the moment is there is no current treatment for COVID-19. That will come soon and hopefully very soon, okay? So before going to questions, just a reminder that we have some other upcoming webinars and training. So 22nd of April, we have a pharmacovigilance talk from Sonia Lopez. And then on end of April, I will be doing a couple of talks about advanced therapy medicinal products. So make sure you register for that, and you can also check them out on YouTube as well when it's all up and running. So with that, thanks very much, and please send us any questions by email or comments in the YouTube, or you can also give us some comments now if you like, and you're connected to the webinar. So please go ahead. Yeah. Is anyone brave? No questions? Sounds like everyone's quiet today. Well, I hope you're all safe anyway. So you can ask questions, as I said, right to Asphalion, or you can put some comments in the YouTube videos and we'll answer those when we get around to it. So thanks very much for watching and listening, and stay safe and stay smart, and we'll see you at the next webinar, which will be in a week's time. Thanks very much.